In botany, the structural diversity of fruits and seeds directly reflects the specific starting anatomy of the flower. Botanically speaking, a fruit is a mature, fertilized, and ripened ovary that develops after fertilization. If a fruit develops exclusively from the mature ovary, it is a true fruit, or eucarp. The mango is a classic example. But when other floral parts, like the thalamus, swell into the fleshy part, it is a false fruit, or pseudocarp. The apple is the primary example of a false fruit. When you eat an apple, the crisp, fleshy material you consume is actually the swollen thalamus. Another major exception is the parthenocarpic fruit. These develop without any fertilization of the ovary at all, making them naturally seedless. The commercial banana is the textbook example. Tracing the development of the ovary wall and the arrangement of the carpels reveals the logic behind these varied botanical structures. As the ovary ripens, its outer wall thickens. This structure becomes the protective layer of the fruit, known as the pericarp. In fleshy fruits, this pericarp differentiates into three distinct anatomical layers: the outer epicarp, the middle mesocarp, and the inner endocarp. This diagram shows a cross-section of a mango, a prime example of a fleshy fruit called a drupe, which develops from a monocarpellary superior ovary. You can identify the thin outer epicarp, the fleshy edible mesocarp, and the stony hard endocarp protecting the seed. Looking at the adjacent coconut cross-section, which is also a drupe, reveals a critical distinction. Instead of a fleshy mesocarp, the coconut features a fibrous mesocarp. Both the mango and the coconut belong to a broader category called simple fruits. By botanical definition, simple fruits are derived from a single flower possessing either a monocarpellary or a polycarpellary syncarpous, meaning fused, ovary. We map drupes along with berries and pomes under the fleshy branch of simple fruits. Conversely, the dry branch of simple fruits contains specimens where the pericarp becomes dry, papery, or woody at maturity, preventing it from differentiating into three distinct layers. This branch divides into dehiscent, indehiscent, and schizocarpic categories. Dry dehiscent fruits split open automatically at maturity to release their seeds. The legume, like the common pea, is the primary dehiscent example. It splits along both the dorsal and ventral sutures. Dry indehiscent fruits do not split open at maturity. Their seeds are released strictly through the natural decay of the pericarp. A caryopsis, such as a grain of wheat or rice, is the standard indehiscent example. Here, the pericarp is completely fused with the seed coat. Finally, schizocarpic fruits act as an intermediate category. At maturity, they break up into single-seeded indehiscent units called mericarps. Whether a simple fruit is a fleshy mango or dry pea pod, they share the exact same origin: a single flower with a fused or single ovary system. Simple fruits arise from a syncarpous ovary, but if a flower has multiple free carpels, it has an apocarpous ovary. In an apocarpous flower, each free carpel develops into a tiny individual fruitlet. The resulting cluster of fruitlets on a single receptacle is called an aggregate fruit, or an iterio. Standard exam examples of aggregate fruits include the strawberry, which is an iterio of achenes, and the custard apple, which is an iterio of berries. Moving up the scale of complexity, we have to look at what happens when an entire inflorescence, a cluster of multiple distinct flowers, fuses together during fruit formation. This large fused structure is defined as a multiple or composite fruit, also known as a syncarp. The pineapple and jackfruit are examples of the cirrhosis subtype, while the fig serves as the classic example of the Syconus subtype. A common board exam trap is identifying exactly which morphological part of a given fruit is actually eaten. To solve this, we can map out an edible parts matrix. Starting with false fruits, recall that for both the apple and the pear, the edible part is the fleshy thalamus. Looking at specific exam favorites, in a lychee, we eat the fleshy aril. In a pomegranate, we consume the juicy seed coat, or testa. And in citrus fruits like oranges and lemons, we eat the juicy placental hairs of the endocarp. Categorizing fruits as simple, aggregate, or multiple, and accurately identifying their edible parts requires looking past their grocery store appearance to identify exactly how many flowers and carpels were involved in their creation. Once you peel away the pericarp, you reach the seed, the mature, fertilized ovule which houses the embryo and its necessary food supply. This image illustrates the structure of a dicotyledonous seed, representing the first major seed classification. Dicot seeds possess an embryo with two cotyledons, with gram, pea, and bean serving as typical examples. The outermost protection is the seed coat, consisting of a thick outer testa and a thin inner tegmen. On the exterior, the hilum is the visible scar where the seed was attached to the fruit wall. Just above it sits the micropyle, a small pore that allows oxygen and water to enter during germination. Opening the seed reveals the internal structure of the dicot embryo. The two cotyledons dominate the interior, typically fleshy and packed full of reserve food materials. Between them lies the embryonal axis. Its upper end is the plumule, which will eventually become the plant's shoot, and the lower end is the radicle, which will become the root. This cross-section shows the structure of a monocotyledonous seed. Monocots possess only one cotyledon, and maize and wheat are the primary study examples. Cereal monocots feature a unique anatomical trait. Their seed coat is membranous and completely fused with the fruit wall. Taking up the bulk of the interior is the massive endosperm, which stores large food reserves. A protein-rich alurone layer separates this endosperm from the embryo. The monocot embryo itself is situated in a distinct groove at the base of the seed. Within this embryo, the specialized, shield-shaped single cotyledon is called the scutellum. Monocots also feature unique protective sheaths. The coleoptile covers and protects the plumule, while the coleorhiza covers the radicle. Regardless of whether a seed is a dicot or a monocot, its ultimate function is identical: to protect the fragile embryonal axis and provide it with enough targeted nutrition to survive the process of germination. The final way to classify seeds for exams is based on exactly where they store their food, either retaining the endosperm or absorbing it entirely into the cotyledons. A comparative matrix makes it easy to lock in these definitions. Endospermic or albuminous seeds are those where the endosperm persists as a permanent food storage tissue. This happens because the embryo does not consume it all during its initial development. Most monocots, like maize and wheat, fall into this category, but notably some dicots, like castor and cotton, are also endospermic. Non-endospermic, or exalbuminous seeds, are those where the endosperm is completely absorbed by the growing embryo. In these seeds, all food reserves are transferred directly into the fleshy cotyledons. Most dicots, such as the pea, gram, and bean, are non-endospermic. However, as a rare exception, some monocots, like orchids, are also non-endospermic. By combining the number of cotyledons with a specific food storage strategy, you can generate a complete morphological profile of any seed. In biological morphology, every classification records a specific chapter in a plant's developmental history. Mastering the terminology, whether it's differentiating the epicarp from the endosperm, the scutellum from a standard cotyledon, or an aterio from a cirrhosis, is simply an exercise in tracking anatomical changes over time. Rely on these functional matrices for edible parts and endosperm classification, and you can confidently navigate the morphology questions on your board and competitive exams. Ultimately, every single complex fruit or seed is just a flower that has successfully fulfilled its evolutionary purpose.